Hello everyone. Today our topic of discussion is non-disjunction in autosomes and allosomes. Let's move on to the presentation. The term non-disjunction refers to an event in which the chromosomes do not segregate properly. So the usually chromosomes are separated, and these chromosomes move to the opposite poles after anaphase. You all know the cell division phases: prophase, metaphase, anaphase, and telophase. So during anaphase, the chromosomes split at the centromere and move to the opposite poles. So this process is known as disjunction. So the chromosomes disjoin at the centromere portion and move to the opposite poles. So in non-disjunction, that process will not be occurring properly. So the non-disjunction process refers to an event in which the chromosomes do not segregate properly. The non-disjunction may be caused by an improper separation of homologous pairs in a bivalent in meiosis, or a failure of the centromeres to disconnect during mitosis. So the non-disjunction may occur during mitosis or meiosis. So during meiosis, non-disjunction can be seen as an improper separation of homologous pairs. So homologous pair segregation or separation will be improper. Then during mitosis, the failure of centromeres to disconnect during this process. So these are the major process occurring in non-disjunction. The non-disjunction can occur in both autosomes and allosomes. Autosomes are the normal somatic chromosomes. Then allosomes are the sex chromosomes X and Y. So the meiotic non-disjunction usually occur in allosomes because the sex chromosomes X and Y chromosomes are usually dividing with the help of meiosis. Usually germ cells divide by meiosis, and somatic cell divide using a mitotic process. So mitosis occurs in somatic cells and meiosis occurs in germ cells. So the meiotic non-disjunction usually occurs in allosomes, and mitotic non-disjunction occurs in autosomes. Then about non-disjunction allosomes. So allosomes are germ cells or sex cells. So the gametes or sex cells undergo meiotic division to produce haploid gametes. Non-disjunction during meiosis can occur during anaphase of meiosis 1 or meiosis 2. So usually non-disjunction process occur during anaphase. So meiosis occurs in two phases, meiosis 1 and meiosis 2. So we have two anaphases during meiosis. So non-disjunction during meiosis can occur during meiosis 1 or meiosis 2. Then we can divide non-disjunction process into two phases: primary non-disjunction and secondary non-disjunction. So the primary non-disjunction occurs during the first meiotic division. So if the non-disjunction process occurs during the first meiotic division, we call it as a primary non-disjunction. Then all the gametes produced by primary non-disjunction are abnormal. So after primary non-disjunction, the produced gametes are usually abnormal. Then secondary non-disjunction occurs during the second meiotic division or meiosis 2, and after secondary non-disjunction we will be having two normal gametes, one gamete with an extra chromosome and one gamete with a missing chromosome. So here some normal chromosomes will be produced and some abnormal chromosomes will be produced. So conditions like trisomy, monosomy, nullisomy, etc. When a gamete containing an extra chromosome n plus 1 is fertilized by a normal gamete, the resulting individual will be having 2n plus 1 chromosome complement. So usually normal gamete after meiosis is represented by n, and a gamete containing extra chromosome is represented as n plus 1. So when a gamete containing an extra chromosome n plus 1 is fertilized by a normal gamete with a chromosome set n, the resulting individual will have an extra chromosome and have a chromosome can be represented as 2n plus 1. Such condition is known as trisomy. Trisomy here we can see 2n set plus 1 extra chromosome, so 2 plus 1 is equal to 3, so therefore it is referred to as a trisomic condition. Then when a gamete with a missing chromosome, that is n minus 1, so from that haploid set one chromosome is missing, so n minus 1 is fertilized by normal gamete with a chromosome complement n, the resulting individual will have 2n minus 1 chromosome complement. That is, these chromosomal set will be having diploid chromosomes but lacking one chromosome. So 2n minus 1, 2 minus 1 is equal to 1. Therefore, this condition is referred to as monosomy. When an individual have two chromosomes less than the basic number, then it is called as nullisomy. Nullisomy means if chromosomal set is lacking two chromosomes, from that diploid 2n set, two chromosomes are lacking. Therefore, 2 minus 2 is equal to 0. Then we can call it as a nullisomic condition. So, non-disjunction during meiosis 1. So here you can see out of the 4 gametes formed, 2 will be n plus 1 and 2 will be n minus 1. So in that picture you can see 2 red colored chromosomes. So that is not 2, actually we have 46 chromosomes. That 2 chromosomes are just for representation. Gametes are produced from a diploid precursor cell. The diploid precursor cell undergo 2 meiotic divisions to produce 4 haploid gametes. The diploid precursor cell first undergo first meiotic division to produce 2 primary gametocytes. Here the 2 primary gametocytes formed are having an unequal chromosome segregation. That is, one daughter cell carries an extra chromosome and the other daughter cell lack that particular chromosome. So the chromosomal segregation during first meiotic division is improper here. So non-disjunction has occurred during first meiotic division. So the primary gametocytes next undergo second meiotic division to produce 4 haploid gametes. So the daughter cell which carries an extra chromosome produces 2 gametes with an extra chromosome. Therefore, they will have a chromosome complement of n plus 1. That is, their chromosomal set is haploid n plus they carry an extra chromosome. Therefore, we represent their chromosome complement as n plus 1. And the other daughter cell, primary gametocyte, also divides to produce 2 haploid gametes, and they lack that particular chromosome, and the 2 haploid gametes produced from this primary gametocyte will be lacking a particular chromosome. So they will have a chromosome complement of n minus 1. That is, they are having a haploid set n minus, they are lacking 1 chromosome. So they are represented as n minus 1. So when the n plus 1 gametes are fused with a normal gamete, they lead to the production of trisomic individuals. That is n plus n is normal gamete, n plus 1 is an abnormal gamete with an extra chromosome. So they'll, when these two combine, they'll produce trisomic condition, 2n plus 1 condition. And when the gametes having n minus 1 chromosome constitution combine with a normal gamete, they will produce progenies with monosomic condition. Monosomy can be represented as 2n minus 1. Therefore, non-disjunction in meiosis 1 leads to trisomy or monosomy. Non-disjunction during meiosis 2. Here the first meiotic division is proper, and the precursor diploid cell produces two haploid daughter cells with normal chromosomal complement, and this primary gametocytes undergo second meiotic division and produces 4 haploid gametes. So here the non-disjunction process occurs during meiosis 2, and then the top primary gametocyte divides and produces 2 gametes, one carrying an extra chromosome, that is having n plus 1 chromosome complement, and the other daughter cell lacking that particular chromosome, that is n minus 1 chromosomal complement. And the bottom portion primary gametocyte produces 2 daughter cells with normal chromosomal complement, because non-disjunction is not taking place in that bottom primary gametocyte. So after second meiotic division, we will get n plus 1 gamete, n minus 1 gamete, and 2 normal gamete. So in meiosis, in non-disjunction during meiosis 1, you won't get normal gamete, all will be abnormal, n plus 1 gamete and n minus 1 gamete. But during meiosis 2, when non-disjunction takes place, you will get 2 normal gametes. When the n plus 1 gamete fuses with a normal gamete, it will produce trisomic progenies, that is 2n plus 1. And when the n minus 1 gamete fuses with normal gamete, it will produce monosomic progenies having 2n minus 1 chromosome complement. And when the normal gamete fuses with another normal gamete, you will get normal diploid condition, that is 2n. Therefore, non-disjunction in meiosis 2 leads to trisomy, monosomy, and normal diploids. So you won't get a normal diploids if non-disjunction has occurred during meiosis 1. Moving on to non-disjunction in autosomes. So autosomes are chromosomes which control somatic characters, and from a single cell, billions of cells are formed in an adult human being by mitosis. So mitosis is also a cell division process which helps to divide normal somatic cells, so they proliferate and regenerate the normal cell population. So many cells like skin cells are continuously replaced by mitosis. So it is a normal everyday process in our body, mitosis. Mitotic non-disjunction refers to the inability of chromatids to separate or disjoin during anaphase. So mitotic non-disjunction can be defined as the inability of chromatids to separate or disjoin during anaphase. Normally chromosome should be moving to opposite poles, breaking at the centromere part. So that segregation is not proper in mitotic non-disjunction. And this mitotic non-disjunction may arise due to several factors like improper segregation of sister chromatids, so that one daughter cell has 3 copies of a chromosome whereas the other daughter cell has only 1. So mitosis usually produces diploid cells, that is from a diploid mother cell it will produce diploid daughter cells. So normally after mitosis daughter cell will carry to an condition, diploid condition, they will carry 2 copies of each chromosome. If non-disjunction is taking place during mitosis, one daughter cell produced will carry an extra chromosome having chromosomal complement 2n+1, and the other daughter cell which lack that particular chromosome will be having 2n-1 chromosomal complement. Then another factor which leads to non-disjunction is improper attachment of chromatids to the spindle fibers, so that it does not migrate to a pole. So spindle fibers are some fiber-like structures made up of cytoskeletal proteins. So these proteins attach to centromere of chromosomes and pull the chromosome to opposite poles during anaphase. So that if that spindle fiber attachment is not proper, the migration of chromosomes to opposite pole will be abnormal. Then another factor is failure of the centromeres to disconnect during mitosis. So centromere should break and disconnect or disjoin and move to the opposite poles, so that this disconnection or disjoining is not occurring properly. And non-disjunction in mitosis, that is, you can see a diploid mother cell, and that diploid cell undergo mitosis to produce 2 diploid daughter cells. And if non-disjunction has occurred, one daughter cell will carry an extra chromosome and have 2n+1 chromosomal complement, and the other daughter cell produced will be lacking that particular chromosome and have 2n-1 chromosomal complement. So mitosis is usually used for proliferation of somatic cells. So all the somatic clone of 2n-1 cell will produce somatic clone of monosomic cells, and all the clones produced from 2n+1 cell produces somatic clone of trisomic cells 2n+1. So 2n-1 condition is known as monosomy, 2n+1 condition is known as trisomy. So in mitosis, non-disjunction leads to the formation of monosomics or trisomics. Non-disjunction in mitosis may lead to mosaicism. Mosaicism is the presence of different chromosome constitution within the cells of an individual. So an individual carry different chromosomal constitutions in different cells. So nearly 50% with Turner syndrome exhibit mosaicism. Turner syndrome is a chromosomal anomaly syndrome, and the persons with Turner syndrome will be having 44 chromosomes plus only one sex chromosome, that is X. Normally a person will be having 46 chromosomes, that is 44 autosome plus 2 6 chromosomes X and Y. Here these persons will carry 44 chromosomes, autosomal chromosomes, plus 1 6 chromosome, that is X. They are lacking Y chromosome, and also it does not possess an extra X chromosome. So the persons which exhibit Turner syndrome may exhibit mosaicism. Some cells are normal in these individuals with which possess an extra X chromosome, and the other cell lack an extra X chromosome. So they are a mixture of 46 chromosome with 2 X chromosome, and other mixture of cell possess only 45 chromosomes in total. That is, they carry only one X chromosome. So we call it as call this phenomenon as mosaicism, because they are having 46 plus XX and also 45 plus X cells. The mechanism of non-disjunction. In normal meiosis, homologous chromosomes pair up, and prior to the first meiotic division, the members of each homologous pair migrate to opposite poles of the cell by means of the pulling action of spindle fibers. So during normal meiotic division, homologous chromosomes will pair up. We call this pairing of homologous chromosome as a synapsis. And prior to the first meiotic division, the members of each homologous pair migrate to opposite poles. So the homologous chromosome migrate to opposite poles of the cell, and this migration is usually brought about by the action of spindle fibers. Those spindle fibers pull these homologous chromosomes to opposite poles. So upon completion of meiosis, each gamete will contain one copy of every chromosome. So after meiosis, each gamete will be carrying only a copy of every chromosome. If two homologous chromosomes fail to separate or disjoin, both migrate to the same pole and give rise to two types of gamete. So if the segregation is not proper and the homologous chromosome fail to separate or disjoin, both the chromosomes migrate to the same pole, and this gives rise to two types of gamete. The one type of gamete possesses two copies of the same chromosome, whereas the other type lacks that chromosome altogether, resulting in aneuploidy. So one gamete contains two copies of the same chromosome, whereas the other gamete lacks that particular chromosome. This will result in aneuploidy. Aneuploidy we have already discussed. Fusion of an aneuploid gamete with a normal gamete gives rise to a zygote with an odd number of chromosomes. We'll discuss some examples from humans. The non-disjunction creates several chromosomal anomalies, and we'll discuss some of the chromosomal anomalies arising after non-disjunction. First one is Down syndrome or trisomy 21. Down syndrome is caused by the presence of an extra copy of chromosome 21. So we usually will have two copies of each chromosome. So here we'll have an extra copy of a particular chromosome, that is our chromosome 21. When the chromosome 21 is present in three copies, that is termed as trisomy 21, and trisomy 21 is the characteristic feature of Down syndrome. Majority of the cases results from primary non-disjunction during maternal meiosis 1. So Down syndrome usually arises after primary non-disjunction during maternal meiosis 1. Primary non-disjunction means that non-disjunction occurs during meiosis 1, and especially this Down syndrome arises after primary non-disjunction during maternal meiosis. Maternal means the ovum. During the cell division of ovum, non-disjunction occurs in the first meiotic phase. And the Down syndrome individuals carry a total of 47 chromosomes because they carry an extra chromosome of 21st chromosome. People with Down syndrome show variable degrees of mental retardation with an average IQ about 50. So Down syndrome patients will show variable degrees of mental retardation and their IQ will be very less. Many people with Down syndrome also have characteristic facial features. They will show evident facial features. Then some retardation of growth and development and an increased incidence of heart defects, leukemia and other abnormalities. So the Down syndrome individuals will have several characteristic facial features, retardation in growth and development, increased risk of some diseases, heart defects, leukemia, etc. Klinefelter syndrome. Klinefelter syndrome is one of the most common 6-chromosome aneuploidy in humans. Most cases are caused by non-disjunction errors in paternal meiosis 1. Here, the non-disjunction occurs during paternal meiosis 1. Individuals with this syndrome have one extra X chromosome, resulting in the karyotype XXY. Individuals with Klinefelter syndrome will carry one extra X chromosome, resulting in karyotype XXY. Here, the fusion of an XY sperm with a normal X egg or the fusion of a Y sperm with an XX egg gives rise to an XXY individual. Usually we will have combination of XX or XY. Here we will get a combination of XXY. This arises due to non-disjunction. Individuals affected by this disorder usually have below average intelligence. Phenotypically, these individuals are male because they carry a Y chromosome. If an individual carry Y chromosome, surely that individual will be a male. So, Klinefelter patients are phenotypically male but present some female secondary sexual characteristics. As they carry 2 copies of X chromosomes, they will also present many secondary sexual characteristics of female. They are often taller than normal and may show gynecomastia. These are breast enlargement in males and are sterile. These are the major features of Klinefelter syndrome. Turner syndrome. This is another chromosomal disorder. An individual having one chromosome less in its diploid complement is called monosomic. Monosomic individuals carry one chromosome less than its normal counterpart. Non-disjunction of the 6 chromosomes in man results in a monosomic condition XO. While the 6 chromosome is undergoing meiosis, non-disjunction may take place and leads to a monosomic condition XO, because that means they does not carry an extra X chromosome or they does not carry even a Y chromosome. These individuals are viable but sterile females. As these individual does not carry a Y chromosome, they will be surely they will be females, but they are sterile. They have 44 autosomes and only an X chromosome. These individuals carry 44 autosomes plus 1 X chromosome. Physical characteristics of the Turner syndrome include short stature, webbing of the neck, broad shield-like chest. So these are some characteristic features of Turner syndrome. The eyes are abnormal and the ears are low set on the head. Their eyes are abnormal and ears are low set on the head. Next one is trisomy X. Trisomy X is a form of 6 chromosome aneuploidy where females have 3 instead of 2 X chromosome. Usually a female will have 2 X chromosome But here a female individual will have 3 copies of X chromosome, resulting in 47 chromosome. Most patients are only mildly affected by neuropsychological and physical symptoms. So most patients show neuropsychological and physical symptoms, and the effects are mild. About 60% of cases were caused by non-disjunction in maternal meiosis 1. So majority of the cases arises due to maternal meiosis 1. Next one is trisomy 18 Edwards syndrome. About 80% of the infants with Edwards syndrome are female. Individuals with trisomy 18 are small at birth and have multiple congenital malformations affecting almost every organ in the body. Those individuals or infants will have several congenital malformations. Clenched fist, an elongated skull, low set malformed ears, mental and developmental retardation, and many other abnormalities are associated with the syndrome. Trisomy 18 is caused by a meiotic non-disjunction event. With non-disjunction, a gamete is produced with an extra copy of chromosome 18. The gamete thus has 24 chromosomes. So here the Edwards syndrome arises after the non-disjunction of chromosome 18. So this condition is known as trisomy 18. Those individuals will possess extra copy of chromosome 18, and they will carry 3 copies of chromosome 18, and the gamete will during the gametogenesis process you will get a haploid gamete, and a haploid gamete usually carries 23 chromosomes. Here after the non-disjunction of chromosome 18, you will get a gamete with 24 chromosomes. So when that gamete with 24 chromosome with a extra copy of chromosome 18 fuses with a normal gamete, that will lead to production of trisomy 18 individuals. When combined with a normal gamete from the other parent, the embryo has 47 chromosomes with 3 copies of chromosome 18. So when the trisomic chromosome 18 gamete fuses with a normal gamete, the embryo formed will possess 47 chromosomes with 3 copies of chromosome 18. Next one is trisomy 13 or Petau syndrome. The characteristics of individuals with trisomy 13 include cleft lip and palate, small eyes, polydactyly. Polydactyly means those individuals will possess extra fingers and toes. Then mental and developmental retardation and cardiac abnormalities. Most infants die before the age of 3 months. So it is a severe chromosomal abnormality. Trisomy 13 is caused by a non-disjunction of chromosomes during meiosis. That is here 13th chromosome will be present in 3 copies. Like all non-disjunction conditions such as Down syndrome, Edwards syndrome, etc., the risk of this syndrome in the offspring increases with maternal age pregnancy. So the probability of production of abnormal gametes will increase with the increase in maternal age at pregnancy. So that's all about non-disjunction in autosomes and allosomes. So out of the chromosomal abnormalities, the most important are Down syndrome, Klinefelter syndrome and Turner syndrome. Thank you all for listening.